Module 3, Part 3. In the first two parts of this module, we have looked at secretory protein synthesis, targeting, and modification of them in the ER and Golgi apparatus. In this part, we will discuss how the proteins are transported out of the Golgi to organelles and protein transport across membranes. So in this part, we are going to talk about what happens beyond Golgi apparatus. In this part, we will focus on Golgi to cell surface trafficking and then Golgi to lysosome trafficking. So with Golgi to lysosome trafficking, so lysosomes are small membrane-bound organelles in the cell. They range in the size from about 0.05 to 0.5 microns, depending on the cell type. You can think of them as bags of hydrolases in the cells that degenerate macromolecules in a regulated fashion. So the interior of the lysosome is filled with a number of hydrolases, such as nucleases, proteases, glucosidases, and lipases, and so on. Here is a list of general types of hydrolase that you would find in the lysosome right here. What is interesting about these hydrolases is that they are all acid hydrolases, which means that the optimum pH for their activity is in the acidic range. So it should come with no surprise that the lysosome lumen or the interior of the lysosome is somewhat acidic compared to the cytosol surrounding it. The pH of the internal lysosome is pH 5 compared to the outside pH, which is 7.2. The pH of the lysosome is maintained by active transport of protons, as shown here, from the cytosol to the lumen of the lysosome by an ATP-dependent V-class pump. We did not talk much about the V-class pump when we talked about the transport of ions across the membrane, but you know that they transport protons. And we are not going to consider this V-class pump in this course, but just to know that they are proton pump, their function is proton pumping, and V-class pump take care of this pH maintenance. So you might be wondering that how is it possible that the membranes of the lysosome stays intact with all these hydrolases, especially with these hydrolases that can degrade lipid because they have lipases. How is it possible that the membrane of the lysosome remains intact? As it turns out, there is a very dense population of glycoproteins in the membrane that I have drawn here, right here. They are only drawn in this one segment, but as you can imagine that the entire membrane is filled with these glycoproteins. So it is thought that these glycoproteins protect the membrane from hydrolysis by the hydrolases within the lumen of the lysosome. Other interesting thing is that the cell is doubly protected from these hydrolases so that these hydrolases don't randomly degrade macromolecules in the cell. First, these hydrolases are enclosed in the lysosomal membrane and so that they compartmentalize these biochemical reaction. It protects the rest of the cell from these hydrolases, which could be detrimental. The second way which these cells are protected is their dependence on the acidic pH for their activity. For example, if there's a breach in the lysosomal membrane and you get some of these hydrolases spilling out into the cytosol, you are now at pH 7.2 and they just don't work very well at pH 7.2. So the cell is doubly protected from these hydrolases and the membrane of the lysosome itself is protected from the hydrolases by this dense population of glycoproteins. In plant and fungal cells, there is a large organelle known as a vacuole, and these can occupy as much as 90% of the cell volume. We sometimes like to call these vacuoles versatile lysosomes. They function like lysosomes in the degradation of the macromolecules, but they also serve as a storage organelle for waste and nutrients. In the case of a plant cell, they regulate the turgor pressure that is necessary to maintain the structure of the plant cell. So the plant and fungal equivalent of the mammalian lysosome is the vacuole. So you can think about the lysosome as the functional hub of which many cellular trafficking pathways. So just for a fun example, that if you fly Delta, you usually fly through Atlanta or Detroit because they are the hubs. In the same way, you can think of the lysosome as a functional hub. So the secretory pathway that we have been talking about includes the lysosome. So you have a synthesis of proteins in the ER, processing in the Golgi, and trafficking to the lysosome. There are two endocytic pathways, pinocytosis or fluid phase endocytosis and phagocytosis, that also traffic into the lysosome. And we will talk about these endocytic pathway in the next part of this topic. There's another pathway known as autophagy, which is the mechanism by which the cells degrade spent organeller components within the cell. Often the portions of the ER need to be recycled and can be trafficked to the lysosome by this autophagy pathway. We're not going to consider the pathway in this course, but it is important to know that the lysosomes participate in a lot of vesicle trafficking pathways, or they are terminal compartment for a lot of vesicle trafficking pathways. So we are talking about trafficking proteins from the Golgi apparatus to the lysosome, and you can see that this is indicated by this large red arrow. So how does the cell know which protein to send to the lysosome by vesicle trafficking and which protein to perhaps traffic to the cell surface from trans-Golgi network? As it turns out that all lysosomal hydrolases, all proteins that are destined for the lysosome, have a specific modification on them. A mannose 6-phosphate, or which is in short known as M6P. So the specific phosphorylation of mannose residues on lysosomal hydrolases is what provides this signal for the hydrolase to move from Golgi apparatus to the lysosome. And the signal is added by an enzyme that is found in the Golgi apparatus known as N-acetylglucosamine phosphotransferase. So here is how it works. There are two sites in the enzyme. One is a recognition site and one is the catalytic site. Here is the lysosomal enzyme, and the lysosomal enzymes will bind in the recognition site, and the catalytic region of this isosome will catalyze the transfer of a phosphate group to the mannose residue on an N-linked glycan on the lysosomal enzyme. This little red square is N-acetylglucosamine that is phosphorylated. So it actually catalyzes the transfer of this phosphate groups to this sugar, this N-acetylglucosamine, to carbon atom 6 of the mannose residue on the N-glycan, which is denoted by this branched structure right here. The reason this modification happens on the lysosomal hydrolases and not on other proteins that they are synthesized and processed through the secretory pathway is because all lysosomal hydrolases have what is known as a signal patch, which is a series of protein sequences found throughout the enzyme polypeptide chain, which is bound specifically to the recognition site of the enzyme. The signal patches have two general motifs. One is based on 2-leucine or dileucine based, which could be either DXLL, X could be any amino acids, else are the leucine, or DEXXLI, something like this. The other signal patch could be tyrosine based, which is tyrosine Y, and then any amino acid, and this phi actually shows that could be an hydrophobic amino acid. So these are the signal patches that are found. So once your protein is modified by this phosphate group and N-acetylglucosamine, there is a phosphodiesterase within the Golgi that will remove the sugar. Now you have this signal, this mannose 6-phosphate or M6P signal, which is then going to direct this lysosomal hydrolase specifically to the lysosomes. This is another rendition of the process. The N-acetylglucosamine phosphotransferase adds the phosphorylated N-linked glycan to the mannose residue of the lysosomal hydrolase. The phosphodiesterase then removes the N-linked glycan, and the result is a lysosomal hydrolase that is tagged with the M6P and destined to the lysosome. This is what it looks like. It turns out there are receptors in the Golgi apparatus that specifically binds to mannose 6-phosphate. These are called the mannose 6-phosphate receptors and shown here. So here is a lysosomal hydrolase with its mannose 6-phosphate tag, and it will bind specifically to these mannose 6-phosphate receptors. So here is a bud that is being formed from the Golgi apparatus. It is a transport vesicle being formed, and you can see that this receptor gets caught up in this bud and it is going to be trafficked to the lysosome. Now you notice that this bud is called the clathrin-coated bud. The reason for that is because that it has a coat of proteins that is made up of clathrin and other proteins. We'll talk about this clathrin coat is so important in trafficking in the last part of the topic. For now, I will mention it because it helps in the formation and or provides the mechanical forces to form the bud, but we will look at this a little later. But at this point, it is safe to say that this bud forms, it has a clathrin coat on it, a protein coat on it. And there is a mannose 6-phosphate receptor bound to the lysosomal hydrolase, and it gets caught up in this vesicle. So then the vesicle then buds from the Golgi apparatus to form a clathrin-coated vesicle, and the vesicle becomes uncoated. So this protein coat disassembles, so now you have a naked vesicle that contains the mannose 6-phosphate receptor bound to the lysosomal hydrolase. So the naked vesicle just have the phospholipid bilayer as a membrane, but there is no protein coat on top of that membrane. What is this vesicle that does is that it fuses with what is known as the late endosome. The disassembly of the protein coat is necessary for this fusion event to take place. It is necessary for a very close interaction between the membranes of the vesicle and the membrane of the late endosome so that the fusion event can happen. If the coat remains, then you can't get interaction between those two membrane lipids and get the fusion event. Within the late endosome, there are phosphatases that will remove the phosphate group from the mannose 6-phosphate. Then that protein is trafficked also by the vesicle trafficking back to the lysosome. What you will notice is that the components of the clathrin coat, the clathrin and other proteins are recycled, shown here in 2A, recycled back to the vesicle budding of the Golgi apparatus. Additionally, mannose 6-phosphate receptor itself is recycled back to the Golgi apparatus as shown here to pick up more of the lysosomal hydrolase. So you remember that when we talked about the ER to Golgi trafficking, we talked about the ER resident proteins must remain in the ER, but occasionally they relocate to Golgi, and through the Cadell receptor, they are returned to the endoplasmic reticulum. So there is a safety net that the cells have. And this is also a similar, this is not a safety net, but this is basically a retrograde transportation of the receptors. But what happens if a lysosomal hydrolase is accidentally secreted out of the cell? The cells also has a safety net to retrieve the lysosomal hydrolase, and it turns out there are mannose 6-phosphate receptors on the plasma membrane. So if there's a lysosomal hydrolase that is accidentally caught up in the constitutive reaction pathway, right here, this is the constitutive reaction pathway, means the hydrolases are going straight onto the plasma membrane rather than going through the clathrin-coated pit into the late endosome and lysosome. Then it can be captured by the mannose 6-phosphate receptors on the plasma membrane into clathrin-coated pit that is forming on the plasma membrane, and then get a clathrin-coated vesicle, uncoating of the vesicle, which fuses with the late endosome, and finally the lysosomal hydrolase is trafficked back onto the lysosome itself. You can see that the cell has been able to retrieve the hydrolase that is accidentally secreted. You can imagine that it would be very detrimental to allow the hydrolases to be secreted into extracellular space because they could be very damaging to the extracellular space. Even though the pH of the extracellular space is not acidic, these hydrolases work somewhat at neutral pH as well. Components in the extracellular space and on the surface of the cell can be destroyed if there isn't some way to retrieve these hydrolases that are accidentally secreted. So trafficking to the lysosome is really important, and there is a group of genetic diseases that are due to either absence of one or more of these lysosomal hydrolases or missing targeting of all lysosomal hydrolases. There are about 40 of these genetic diseases. They are known as lysosomal storage diseases. The reason they are called storage diseases is because if lysosome is missing certain hydrolases, certain glycosylases, certain lipases, then the macromolecules that are trafficked to the lysosome for degradation don't become degraded because the lysosomal hydrolases are missing. So they accumulate or become stored in the lysosome, And that is why they are called the storage diseases, because they're characterized by accumulation of undigested macromolecules in the lysosome. So I mentioned that there are about 40 of these genetic storage diseases, and here are 3 of them. We will skip over the ICEL disease for now. It is a little bit different, but one you have probably heard of is the Tay-Sachs disease. In the Tay-Sachs disease, these individuals are missing an enzyme, hexosaminase A, and in this case, you have an accumulation of ganglioside GM2 in the lysosomes, which in normal cells is degraded. The outcome of accumulation of ganglioside GM2 in these cells is intellectual impairment, enlarged liver, some abnormalities of the skeletal system, and this disease is often fatal by age 2. There is another you may have heard of, is the Gaucher's disease. In this case, these individuals are missing an enzyme, glucocerebrosidase, resulting in accumulation of glucocerebroside in lysosomes. And in this case, the disease is characterized by liver and spleen enlargement, erosion of bone, and intellectual impairment. So in these 2 examples, Tay-Sachs and Gaucher's disease, you are missing a single lysosomal hydrolase, which has detrimental effects on these patients. Another one, known as the ICEL disease, what is interesting about ICEL disease is that you are not missing a particular hydrolase, but if you have a defect in a trafficking of all hydrolases to the lysosome. It is listed as a storage disease, but it is not truly a storage disease because you don't have the accumulation of macromolecules in lysosomes. But the real detrimental symptom in this disease is that you have severe tissue destruction because those hydrolases which don't get traffic to the lysosome actually end up in the constitutive secretion pathway and end up outside the cell. So here is the problem with the ICEL disease. An individual with an ICEL disease have a non-functional N-acetyl-glucosamine phosphotransferase. And you will remember that this enzyme that actually catalyzes the mannose-6 phosphate modification on lysosomal hydrolases. So in these individuals, their lysosomal hydrolases do not have the mannose-6 phosphate. And because of that, they are not bound by the mannose-6 phosphate receptors, so they are not trafficked to lysosome. Instead, they actually end up outside the cell, and that is why you get this severe tissue destruction. What is really intriguing about the ICEL disease is that these patients in their liver cells and their hepatocytes have completely normal distribution and targeting of lysosomal hydrolases to lysosomes. So even without the mannose-6 phosphate modification, there is some different targeting mechanism in cells as seen in this case. These liver cells that can get lysosomal hydrolases to lysosome without the mannose-6 phosphate modification. As of yet, that pathway has not been determined completely, but the scientists are working hard with the proteomics to define that pathway, but so far we do not know. Likewise, there are some lower single eukaryotes that don't put mannose-6 phosphate modifications on their lysosomal hydrolases, but the lysosomal hydrolases get to the lysosome just fine. So these lower eukaryotes are being used as a model to discern these alternative targeting pathway for the lysosomes. So that's the Golgi to the lysosome pathway. Let's talk about the Golgi to the cell surface pathway. So in every single eukaryotic cell, there is a stream of vesicles that are budding off from the trans-Golgi network and moving to the plasma membrane and fusing with the plasma membrane. This is known as the constitutive secretion pathway, and this pathway is very important and critical to cells because it is the pathway by which plasma membrane proteins are delivered to the plasma membrane and the process by which the components of the extracellular space are released. Now, there is a subset of cells that also possesses what is known as a regulated secretion pathway. In this case, you have vesicles budding from the trans-Golgi network that contain a cargo that is going to be secreted out of the cells in a very regulated way. So not constitutively released from the cells, But when the cell receives a signal, so not constitutively released from the cells, but when the cell receives a signal, in this case, a signal such as a hormone or neurotransmitter, these secretory vesicles fuse with the plasma membrane, releasing their content. So cells that make hormones, cells that make neurotransmitters, cells that synthesize digestive enzyme, all of them have this regulated secretion pathway. So in these cells, you can see that these secretory vesicles that have been budded from the trans-Golgi network sitting just right below the plasma membrane, waiting for a signal to arrive, and then they will fuse with the plasma membrane to deliver their cargo into the extracellular space. So here is a couple of examples. This is a transmission EM of a pancreatic cell. These possesses the regulated secretion pathway, and this pathway by which the insulin is released from these cells. Here you can see a secretory vesicle sitting right below the plasma membrane, and here you can see the secretory vesicle just fused with the plasma membrane. This very dark electron dense structure that you're seeing, that it is insulin because it is a protein. It takes up the negative stain very rapidly. Sometimes the secretory vesicles are called the secretory granules or dense core granules because the contents of them are usually electron dense, as you can see here, so that they are very dark and very distinguishable by electron microscopy. So here the question is that what is the trigger for the insulin? As you know that blood sugar, when the blood sugar level goes up, there are receptors for that, and that increase of the blood sugar level release the insulin into the blood. Here is a dramatic example of this regulated secretory pathway. This is a transmission EM of a rat mast cell. And you may have probably heard about the mast cells in the immune system, and these are responsible for releasing histamine. They trigger allergic reactions. So the mast cells are one type of white blood cells though. You take Claritin, Benadryl, Zyrtec to stop the release of histamine from the mast cells. So now you know that where your anti-allergy medication work. So here is a mast cell that is not stimulated, and you can see that it is packed with these secretory vesicles that have these dark content, which is the electron-dense histamine. Here is a mast cell that is stimulated where a number of secretory vesicles have already fused with the plasma membrane, and this transmission EM caught this cell right at the moment of a fusion. So you can see that very dramatically how all these secretory vesicles have fused with the plasma membrane and in the process of being fused. So this is an active stimulated mast cell releases the histamine, which will trigger allergic reactions. In the previous part right here of this topic, we talked about a number of vesicle trafficking pathways through the secretory system of the cell. So we have already looked at endoplasmic reticulum to Golgi trafficking, Golgi to lysosome trafficking, Golgi to cell surface trafficking. The last vesicle trafficking pathway I want to talk about in this topic are the endocytic pathway. That is shown here as the red arrow. And this is the movement of substance and macromolecules, large particles, from outside of the cell to the inside of the cell, sometimes small particle as well. So we have already talked about three endocytic pathway to lysosomes, and we mentioned that the lysosomes are functional hub, and they are the endpoint of a number of endocytic pathway in eukaryotic cells. And these pathways include phagocytosis, which is uptake of large particles right here. Endocytosis or fluid phase endocytosis, which is the uptake of small particles, food or sometimes receptor from the extracellular space or the plasma membrane. And there is autophagy, which is the process by which the cells can direct the transport of spent and old organelle like this spent mitochondria or the old mitochondria for degradation and recycling of the components. We're not going to consider this autophagy in this course. In this part, we are going to talk about the phagocytosis and the fluid phase endocytosis, which is also known as pinocytosis or pinocytosis. So let's start with phagocytosis. So as I mentioned, it is the process by which large particle as large as bacteria can be ingested into by the cell. Phagocytosis is very common among lower cell eukaryotes like amoeba or protozoa. In these type of cell, phagocytosis is carried out to obtain nutrients. So it is for feeding, and that is a really important function. In multicellular organism, the phagocytosis is somewhat rare, and it is carried out by a subset of cells known as professional phagocytes. And they are the cells that are usually a part of the immune systems. In the blood, you have one type of circulating white blood cells with non-granular cytoplasm known as agranulocyte. One type of agranulocyte is a monocyte. The other type, this is not needed, just an extra. The other types are called the lymphocytes, which are the T and B cells. Now, this monocyte leave the circulation and move into the tissue spaces, and now they are called the macrophages. These are responsible for recycling other cells and also phagocytose pathogens like bacteria. On the other hand, there are three types of white blood cells or leukocyte, which are granulocyte, and neutrophil is one of them. And neutrophil also circulates in the blood, but often leave the circulation and migrate into the tissue space looking for invading pathogens. The purpose of phagocytosis in multicellular organism is usually an immune response to invading microorganisms, and often it is used for scavenging of old or damaged cells. So there are two really important points we want to know about phagocytosis. First of all, it requires a surface receptor. So the phagocytosis does not happen randomly on the plasma membrane. A phagocytic target must bind to a specific receptor on the cell surface in order to be taken up by this pathway. A second important feature of phagocytosis is that it is a trigger or an induced event. So it is not continuous random uptake of particles from the extracellular space. What I mean by triggered is that once a phagocytic target binds to its receptor on the cell surface, that triggers the cell or induces the cells to extend its membrane-forming pseudopods around the particles that is being ingested. So it is a triggered event. So here is an example of phagocytosis. Now the antibodies protect us by binding to the invading bacteria. When antibodies specifically bind to an infectious agent such as a bacteria, the antibodies are oriented in such a way that the variable region will bind specifically to the bacterium, but the Fc portion or the stalk right here will be sticking out. These antibodies will form a coat around these infectious microbes. Well, as it turns out, the macrophages and the neutrophils have receptors on their cell surface that are specific to the Fc portion of these antibodies. So they have the Fc receptors. So what happens is that you have an invading bacterium or bacterial infection. Your body will produce antibodies. These antibodies will bind and coat this bacterium. Then these important cells of the immune system, the macrophages and neutrophils, can bind to the Fc portion of these antibodies. This triggers phagocytosis, uptake, and their destruction. So this is an important immune function. So here is a transmission electron micrograph of a neutrophil that is phagocytosing a bacteria. In fact, we saw this EM image in early topic. Two bacteria shown here, one here, one here. These large membrane extension here from the neutrophil right here, these are the pseudopodia or the pseudopods, singular. The neutrophil is stretching its membrane around these bacteria to engulf and phagocytose them into the cell. So here is a scanning micrograph of a macrophage that is phagocytosing some red blood cells that are actually spent or old red blood cells. These red arrows are pointing towards the membrane of the macrophage that is reaching out and reaching around the red blood cell. Macrophage can scavenge by phagocytosis 10 to the power 11th old red blood cells per day in each of us. This is a really important function for the macrophages to recycle the red blood cells. So let's look at other form of endocytosis, and that is the fluid phase endocytosis, and sometimes referred to as pinocytosis. As I mentioned earlier, that the fluid phase endocytosis is the way that small particles and fluid can be taken up from the extracellular space. Sometimes the cell surface receptors are also taken up, and when this happens, the process is called receptor-mediated endocytosis. We'll talk about that in a minute. A major difference between the fluid phase endocytosis and phagocytosis is that the fluid phase endocytosis
is a continuous process. This is very important. It is non-specific, so the cell is constantly carrying out fluid phase endocytosis. There is no extracellular event that triggers the cell to form a fluid phase endosome. It is just a continuous, non-specific process. The rate depends on the cell type. For an example, a macrophage can take about 25% of your entire volume in fluid phase per hour. But some lower eukaryotes, some lower single-cell amoeba like Dictyostelium and Entamoeba can take up greater than 75% of their entire cell volume in fluid per hour. So lower eukaryotes are very robust pinocytic cells, and these lower eukaryotes use fluid phase endocytosis for taking up nutrients. So fluid phase endocytosis begins at a very specialized region of the cell known as clathrin-coated pits. These are small invagination of the cell that are coated with a group of proteins that includes clathrin. But the clathrin is not the only protein that is present. There are many other proteins involved in there as well. The protein coat is electron dense, as you can see that you can appreciate from these transmission EMs, but these clathrin-coated pits are very short-lived. Shortly after invagination forms, the invagination extends and they pinch off to form these clathrin-coated vesicles. So as I mentioned, this is a non-specific event in all eukaryotic cells. It happens on many locations on the plasma membrane. In fact, it is thought that clathrin-coated pits occupy about 2% of the total plasma membrane at any given point. And this happening continuously along the plasma membrane. So I also mentioned that sometimes the cell surface receptor can be taken up by these clathrin-coated pits. When that happens, the process is called receptor-mediated endocytosis. So as it turns out, the proteins in the clathrin-coated pit have the propensity to attract cell surface receptors or bind cell surface receptors and trap them into the pit. So cell surface receptors are shown here in the blue. They most look like this Y-shaped antibody, but they are not. Please don't get confused for antibodies. They are usually transmembrane proteins that bind specifically to the macromolecules or ligands in the extracellular space. So these are receptors, not antibodies. So here is a clathrin-coated pit that is being formed right here. You can see that there are some receptors that are found themselves in this pit. If these receptors bind to a ligand, the clathrin-coated pit, it invaginates and will get pinched off. Those receptors and their cargo or ligand, and they will be found on the clathrin-coated vesicle. Now I need to stress that this is not a specific event. It wasn't the receptor in the pit that triggered the formation of the coated vesicle. The coated vesicle would have formed even if there were no receptors in that pit, and this coated vesicle would have formed if there were no receptors in that pit, even if they weren't bound by any extracellular ligand. So it just happens spontaneously. If a receptor finds itself in one of these pits, it gets taken up into the coated vesicles. So here is example of receptor-mediated endocytosis, probably the one best understood, and one of the first pathways that were described in the receptor-mediated endocytosis of cholesterol in mammalian cells. So most mammalian cells carry out receptors-mediated endocytosis to acquire their cholesterol, which is important for building the membranes. And if this process doesn't work, cholesterol builds up in the bloodstream and it seriously damages the blood vessels. You get the formation of plaques, you get arterial disease, high propensity for stroke, heart attack, etc. In fact, much is learned about this pathway by studying a group of individuals who had genetic disorders in which they could not carry out receptor-mediated endocytosis of cholesterol. So these individuals had the signs of heart disease and arterial disease very early in their lives, and they were very, very high risk for sudden cardiac death. And I'll tell you in a minute what those genetic defects were. So in studying these cells, these individuals, we learned quite a bit about the receptor-mediated endocytosis. So most of the cholesterol is carried in these large particles of low-density lipoprotein or LDL particles, which is responsible for delivering cholesterol from the liver to the intestine to all cells in our body. These particles are delineated by a phospholipid monolayer. So this is important. It's not a bilayer, a phospholipid monolayer. Most lipid layers are phospholipid bilayers. The plasma membrane is a phospholipid bilayer, but here is an example of a phospholipid monolayer that we mentioned before as well. So this phospholipid monolayer, there are few molecules of cholesterol shown here in green, but the important part is the cargo. There are about 1,500 cholesterol molecules within the core of this structure. The LDL part is also bound to a protein known as the ApoB protein, and this ApoB protein is the one which mediates or regulates the receptor-mediated endocytosis. It is this protein that interacts with the receptor on the cell surface. So here is a 3D rendition of the LDL particle. You can very clearly see the phospholipid layer and the cholesterol molecule intercalated. Then you can see that there's a very dense core that contains about 1,500 cholesterol molecules. And here is the ApoB bound to the particle. So here is an impressive picture. There are about 0.02 to 0.03 micron in diameter and clearly visible under EM, but without much detail. But the above picture shows the power of cryo-electron microscopy, and the top pane shows the original image right here. And you can easily see that this is the 3D reconstruction of the particle. The bottom picture shows the ApoB bind to the core particle. The different domains of the protein is colored differently though. So here is what the receptor-mediated endocytosis looks like. So in step 1, you have the formation of the clathrin-coated pit, and you will notice that there is an LDL receptor in green, and that has found itself in this clathrin-coated pit that has been formed randomly. Here is an LDL particle in the extracellular space. The LDL receptor binds to the LDL particle, and it's a very tight interaction between the LDL particle and the receptor. Then the clathrin-coated pit invaginates and pinches off and forms a coated vesicle. Shortly after the vesicle forms, it becomes uncoated, and at that point, it is known as an early endosome. The interior of the late endosome is somewhat acidic. It has a pH of 5, and that facilitates the release of the LDL particle from its receptor. So in the acidic pH, the LDL is released from the LDL receptor because at acidic pH, the affinity of the receptors and the ApoB protein, which is present on the LDL particle, is much lower. Then the LDL particle is trafficked to the lysosome, where it is completely degraded, and the cholesterol is used for membrane building. The fatty acids are used for the membrane building, and the amino acids of the ApoB protein are completely recycled as well. Want to point out that this uncoating event is very, very important. So when you go from a coated vesicle to early endosome, it is really important so that the vesicle has the LDL particle in it. Without it, it cannot fuse to the late endosome. So in order for this early endosome to fuse with the late endosome, you have to be very close interaction of the lipids to the membranes. Here is a transmission electron microscopy of LDL uptake. In this case, the LDL used is the LDL ferritin, and ferritin is electron dense because it's iron and appears dark. This is a modified experiment that allows you to visualize this clathrin-coated vesicle, and the coated pit is being formed, clathrin-coated vesicle being formed, and this is bound onto the ferritin, and that's the reason that you can see them as the dark. So ferritin bound to the clathrin-coated pit, bound to the clathrin-coated pit, and taken in. Then what happens with this invagination and pinching off from it, the clathrin-coated vesicle, and the panel D right here shows the LDL ferritin within the early endosome. In about 6 minutes after the pinching of that vesicle, this is actually a closer picture of the early endosome. So I mentioned earlier that there are some groups of individuals that have genetic defects and prevent their cells from carrying out receptor-mediated endocytosis of the LDL particles. Again, this is detrimental because cholesterol accumulates in their blood and can lead to arterial disease, heart attack, stroke, sudden cardiac death, etc. In some individuals, the LDL receptor is missing altogether. And you can see that this is the cell where LDL receptor is missing. In others, there's a genetic mutation in the LDL receptor that causes it to be unfolded, and it is removed from the secretory pathway, but the unfolded protein response that we talked about. So these cells don't have a receptor at all. There's another group of individuals that have a normal number of receptors, but there's a mutation on these receptors that prevents the association of them with the clathrin-coated pit. They have LDL receptors that can bind to their LDL, and the cells form clathrin-coated pit, but the receptors cannot associate themselves in the clathrin-coated pit, so the outcome is the same, that they cannot deliver the LDL to the clathrin-coated pit, to the endosomes and into the cell. So having the receptor and having that mutation not being able to associate with the clathrin-coated pit, the outcome is the same. So we talked about what happened to LDL once it gets into the cell. It is eventually trafficked to the lysosome for degradation. But what happens to the receptor? What is the fate of the cell surface receptors that are taken up by this pathway? There are actually three possible fates for these receptors. The first possibility is that the receptor can be simply recycled back to the plasma membrane, to the same membrane domain which they came from. The second possibility is the degradation, in which they themselves traffic to the lysosome with their cargo to be degraded along with their cargo. The third possibility is a transcytosis, where the receptors are taken up in one domain, but then they are trafficked to the different domain in the cell. So there is an epithelial cell. You will remember that the epithelial cells are polarized and that have different membrane domains: the apical membrane domain and the basolateral membrane domain. These domains are maintained by the tight junctions, which do not allow proteins from one domain move in the phospholipid bilayer and end up in the other domain. The transcytosis in this experiment would be uptake of the receptor on the apical surface and these cells, and then traffic of the receptor to the basolateral side. So let's take a look at some of these examples of all of these. So let's go back to the LDL receptor. In this case, the LDL receptor is recycled back, right here, is recycled back in the same domain which they came from. So again, the LDL receptor is bound to the LDL, taken up by the clathrin-coated pit. The vesicle uncoats, form the early endosome. The early endosome fuses with the late endosome. The LDL particle is sent to the lysosome for degradation, right here. The LDL receptor is trafficked back onto the same domain of the membrane. If this is an apical surface, it goes back to the apical surface. Here is another example of the receptor that is trafficked back to the same domain, but this one is very interesting because in this case, both the receptor and its cargo, the ligand, are recycled back to the same original membrane domain, the cargo as well as the receptor. Very interesting. So here is a receptor-mediated membrane endocytosis of transferrin. Transferrin is a major blood glycoprotein that is responsible for delivering iron from the liver, particularly all cells in the body. And apotransferrin can take on different conformation or forms. When it's not bound to the iron, it is known as apotransferrins, and when it's bound with two irons, it is known as ferrotransferrin. What is interesting is that the apotransferrin and ferrotransferrin have different affinities with the transferrin receptors depending on the pH of the surrounding environment. So in acidic environment right here, the apotransferrin binds very tightly to the receptor, but not in neutral environment. And the opposite is true for the ferrotransferrin. Ferrotransferrin binds very tightly to the receptor at neutral pH, but not very well in the acidic pH. Okay, let's go through this pathway. Here is a clathrin-coated pit, and in that pit is a receptor. Here is a transferrin protein that is bound to 2-iron. So this ferrotransferrin, and it has a high affinity for its receptor at neutral pH in the extracellular space. The extracellular space right here. So it is very likely that the ferrotransferrin will interact with its receptor. So the receptor and its cargo are taken up by the clathrin-coated pit. It becomes uncoated into the early endosome, and then it fuses with the late endosome, which is acidic. So the first thing that happens in the acidic environment is that the iron comes off. So iron comes off and transferred by a transferrin transporter into the cytosol. So now you do not have phytotransferrin anymore. You have all apotransferrin because iron has left this phytotransferrin in this acidic environment. Now remember that the apotransferrin is bound very tightly to its receptor in the acidic environment. The apotransferrin does not come off and its receptor, because it has a high affinity for the receptor, and the receptor and its cargo, which is apotransferrins, are trafficked back to the cell surface. Now the receptor and its cargo are in neutral environment again. Remember, apotransferrin is released from its receptor, so then the apotransferrin can be recycled. Remember, apotransferrin doesn't bind very well to the receptor in the neutral environment, so the apotransferrin is released from its receptor. So then apotransferrin can be recycled, it can pick up more iron, and then enter receptor-mediated endocytosis again. So here is an example of receptor that gets degraded along with its target. It is an epidermal growth factor receptor, or EGF receptor. EGF is an important growth promoting and signaling molecule. When EGF binds the receptor on the cell surface, the cells receive a signal to proliferate. We can talk more about EGF receptor when you talk about the cell cycle, when you talk about cellular basis of cancer and other diseases. Shortly after the signal is propagated, the entire complex, the receptor EGF complex, is taken up by the receptor-mediated endocytosis, and eventually the entire complex is trafficked to the lysosome for degradation. So the third path is transcytosis. This is when a receptor is moved from one domain to another membrane domain. The example I want to use for transcytosis of antibodies across the epithelial cells of the intestine. So the newborn mammals obtain antibodies from their mother's milk, and the antibodies help protect these newborns from infection. And the antibodies are taken up through the transcytosis. Remember that the ingested milk do have these antibodies which do not get digested by the intestinal enzymes because they are glycosylated heavily and they are not degraded. So these antibodies can be taken up by these receptor-mediated endocytosis. So the lumen of the intestine is about pH 6 on this side of the intestine epithelial cells. This is a cross-section of the epithelial cells on the intestines, and you can clearly see that this structure is a tight junction. It maintains the identity of the membrane domains. The other side of the epithelial layer is the blood and the interstitial fluid, which has a pH of about 7. So here are the antibodies that are ingested from the mother's milk. As it turns out, these intestinal cells have the Fc receptors, and they're very similar to the Fc receptors of the macrophages and the neutrophil that are responsible for taking up infectious microbes coated by antibodies. So these antibodies bind to this receptor through their Fc region. They are taken up by the clathrin-coated pit, and they find themselves in a clathrin-coated vesicle and then early endosome. And in this case, they are trafficked to the opposite side of the cell, and they are released into the blood and interstitial fluid region. So this receptor and antibody are taken up on the apical side, and they're trafficked to the basolateral side. So these antibodies will be used to protect the newborn animal. Eventually, these Fc receptors are trafficked back to the apical side of the membrane to be reused right here.
This concludes part 3 of module 3.